Right. So I'm going to talk about something that I would usually say this is a really difficult place to get to. We've not done much research here because it's so far away. There's a huge vertical challenge. I'm going after a talk on Mars, so that now seems pretty minuscule by comparison. But I'm going to talk about the rainforest canopy. So I'll just try and work out how to share my screen. That seems to be there. Let's do that. And now how do I make it a show? Excellent. Right. So I'm going to talk to you about the life epiphytic. So epiphytes and how they exist in the world. I'm actually a research student at the University of Stirling. I'm currently looking at the way that rainforests recover after logging. But just for the sake of this, I wanted to talk about something that was already done and finished as a project rather than something I'm frantically trying to write up into papers as we speak. So that's what I'm going to talk about today. I'm not going to focus too much on the actual research itself. What I want to talk about instead is the methods behind it and why it's all just so fascinating. I assume we've got quite a broad audience today, so I'm going to try and make fun things you can remember as individual facts rather than necessarily delving too deeply into my individual science. We can always talk about that when it comes to questions at the end. So first up, what is an epiphyte? So an epiphyte, the word comes from the Greek, epi meaning on top of, phyte meaning plant. So it's any plant that grows on top of another plant. They make up a huge amount of the plant diversity around the world, and you will almost certainly have seen one somewhere because it includes things like mosses and lichens. So they are absolutely everywhere. There's huge numbers of them. But to narrow it down, what I want to talk about today is vascular epiphytes. So vascular epiphytes are the ones with complex systems used for carrying water and sugars around to different parts of the plants. This wonderful GIF that I've put together and gets rather irritating rather quickly shows just the difference between the tree, the epiphytes living on top of a tree with a couple of examples, and the things Tarzan swings on, which are lianas. And lianas and vines we don't really consider to be epiphytes because they also have contact with the ground during their life cycle. True epiphytes never touch the ground at any point during their lives. So here is a slightly less irritating visual demonstration of what epiphytes look like. Here we can see a load of ferns just wrapped around the trunk of their host tree. And I think you can also see some orchids if you follow the stems of the trees up towards where the camera is. You can also see my rope hanging down about two-thirds of the way along from the right at the bottom of the screen, because this is a very long way up into the canopy. And we will also talk about how we get there. But first I want to discuss probably the most common three families of epiphytes that we run into during tropical studies of vascular epiphytes. So again, these ones with the complex systems of waters and sugars. Also, I'm thinking that these will be some of the little factoids you can take away with you and tell and shock all your friends and relatives. So first things first, we have bromeliads. This actually isn't a picture of a canopy bromeliad because I've not done any work in the neotropics for this kind of thing. This is a photo of a pineapple that I found in my fridge. So they're from a bromeliad family. They don't grow in trees, but many of the things from the same family do, and they all have this characteristic shape of leaves. They're named after Olaus Bromell, who was about and as a Swedish botanist during the time when Linnaeus was coming up with these original binomials. So they're actually named by Linnaeus himself after Olaus Bromell. The next type we've got are ferns. So ferns are the most likely type you'll have seen if you're in a temperate climate at the moment. I'm sure you'll have seen them on trees around about, especially if you're at a place where it will occasionally be covered in fog or cloud cover. So these will grow if there's even a little bit of moisture really in the air to help support them. Ferns are named after fano, meaning their feathered shape. If you think of the more classic ferns, they've got kind of this really nice fractal branching structure. Brilliantly, this comes from a Proto-Germanic, meaning again feathered, porno, which I hope you will tell absolutely everyone you meet. And on a similar theme, orchids, which hopefully you will have seen around somewhere, even if it's just in a pot that someone's keeping as a nice ornamental plant. Orchids come from the term orchis, which is Greek, meaning testicle. And that's to do with the shape of the bulb at the bottom of the leaves. These pseudobulbs allow them to carry a lot of extra nutrients up in the canopy, and is part of how they've got such a successful strategy, meaning that they can flower and fruit and seed, even despite being so far away from the ground floor and most of the nutrients there. So the next question is, why are all these epiphytes important? What's so great about them, and why am I telling you all of this stuff? Well, apart from the fact that they have a huge contribution to diversity, epiphytes are a giant resource for all sorts of other creatures and organisms. So just in terms of the amount of food they can provide, one study, I think in Vietnam, found that epiphytes were providing over 2/3 of all food as a resource being used by birds in that region. So they're hugely important to keeping other things alive. They also provide water, because it means that if you're an animal that wants a drink, you don't have to spend lots and lots of energy climbing all the way down to the forest floor and then climbing all the way up again, if these epiphytes are able to catch water in their leaves. On a similar note, some orchids are able to hold so much water that they actually have these obligate associations with other creatures, and you will sometimes find frogs living their entire life cycles inside bromeliads. So the amount of water they can hold up in the canopy is really important, both for animals that live in them and drink from them, but also for stopping these huge flood events that would otherwise happen much more seriously and much more regularly in rainforests if water was able to fall straight to the forest floor instead of being caught up in the canopy so much. There are various other duties they perform. They give shelter to various other organisms, including ants, which protect their host trees from other predators. And they also change the light environment a little bit. So they create these really complex microclimates and very interesting environments for other things to live in. So they have huge conservation values and these massive knock-on effects, despite being way, way above the heads of us. And it really is a thing that they are far, far above us. So the average rainforest canopy habitat is somewhere sort of around 40 meters high, and the tallest tree in the tropics that was actually only discovered last year, maybe the year before, is over 100 meters tall. So it can be a really difficult thing that if you're in the middle of a rainforest, how do you get up there to study these epiphytes right at the top? So we've been trying this now for pretty much 100 years in various ways and shapes and forms, not me personally, but various different scientists. So the easiest way, as this picture kind of represents in some ways, is using binoculars, just looking up and trying as hard as you can to see what's up there. And it's pretty rubbish because you can't see what's on the top of the branches. You can only see things that happen to be hanging underneath. And even those, if there's any kind of leaves in the way, as you can see in this image, which is really likely there will be, it's quite hard to see what's going on. So this also is true for the absolute high-tech approach, which is flying drones over the top. You can get really, really close with drones now, but you can't get in amongst the individual trees because you keep hitting branches. And the problem with drones is that you can only see the things on top of the branches and you can't see the things lower down the tree. So to find out what's going on up at the top, you need to have a much more comprehensive way of looking. And fairly far in the past now, this was solved with rifles, where you would pick a branch, you would get a marksman, and you would try and break the branch off so it would fall down to the floor by shooting it. And as you can imagine, this is a fairly destructive practice that we don't try much anymore. It goes along with entomologists who, wanting to find out what insects are in the canopy, would just smoke and kill absolutely everything above it and just hope it would fall down onto a tarpaulin they'd laid out, and then spend the next few years collecting and categorizing everything from an individual tree. So there are these very destructive methods that we can use to get things out, but which aren't really used anymore unless it's for a really, really big thing and with very good reasoning, because ethics. On a similar point of ethics, there's also the case that people used to just send up the locals, whoever they might be, because tropical research and tropical exploration, especially canopy research, comes with a lot of baggage from colonialism and many social issues which are very, very difficult to work around their legacy now, and which has a lot of people trying to work to make things better. But in the past, it was very much a case of, we've got this local guide, they can climb up with little to no health and safety checks, and they will bring things back down, which is a huge issue and thankfully no longer happens, at least with people climbing. So the next step really was getting scientists up there. And this is a photograph taken in 1929 from an Oxford expedition, who were the first scientists to properly get up into the canopy themselves and look around. I believe they also had the local mayor in New Guinea who came for a go on the chair and was hoisted up into the canopy, and then the chair broke. So they were stuck there for quite a while, but eventually managed to get down again. But this was really the start of exploring up in the canopy and seeing what was going on. It had really, really promising results, but nothing really happened until the 60s. That's when things actually started taking off in canopy research properly. And this isn't a picture from the 60s itself, this is much more modern, but it's to represent, but in the 60s there was this Dutch expedition to Zaire, where people actually made canopy platforms and spent much longer in much more comfortable positions looking at what was going on, in this case looking at monkeys and seeing how they acted up in the canopy. But nowadays we've got these huge cranes, which mean we're able to very, very carefully examine a lot of what's going on in the canopy. The problem with cranes is they don't move, and they can sometimes be quite destructive to build as well. But the benefit is you get lots and lots of data. So the question is, if you've got a forest canopy like this one, do you want to have one big plot with a crane in it, which is very, very finely detailed, and you're finding lots and lots of information out about it? Or would you prefer all of these different dots dotted around the landscape, where actually you can learn probably a lot less about the individual spot because it's going to be a single tree, but you can get many, many more sites. And sort of 50 years after this expedition to Zaire, that's where I'm coming in with my research. So my research is not based over where that dot just came up in Panama. That's where the majority of research has been done to date. My research is down there in Sulawesi in Indonesia. And the idea behind my master's research was really just fact-checking what happened in Panama, seeing if it still held true on the other side of the globe. So I wanted to find out what kind of trees supported the most epiphytes, so that we could use that information to help with conservation. As I said, I don't want to go into that in too much detail now, but I will use it as a way to talk more about canopy access techniques and how we're doing things in the current day. So this is a map that I wanted to put in mostly because I spent ages making it and then it wasn't allowed to go in the paper that resulted. So I now use it in every presentation I possibly can. A slightly better representation of where we are. So it really helpfully shows we're in this forest just above Australia in the southeast of Sulawesi on the island of Buton. And over about a couple of months in this forest that you can now see on screen, I climbed lots and lots of different trees. Well, about 50. And the way I did it was using these individual points, climbing individual trees with ropes. So as you can see from the GIF that's now kind of working its way through on the screen, you start off with a catapult, you fire a piece of string over the top of your tree, you connect a rope to the other end of the string and pull it through, and then anchoring that rope, you're able to climb the other side of the tree towards, hopefully, success. So the way we climb ropes, and actually the way we shoot ropes, this is half of an 8-foot long catapult. It clips together. and is a very useful thing. These bits of rubber pull down about 8 feet. And then once we've got our ropes, we use little things like this. So if you can see on there, there's lots of little teeth. The rope flows through here, and you're able to grip on and slowly move your way up the rope as you climb. Once you get to the top, you can just descend and start looking at what's actually in the canopy. So the first thing you do when you get to the top of the canopy is obviously you take a selfie. This is me from a few years back now, so I've aged a little bit since then, but I'll also move on quite quickly from this. But the idea of taking the selfie, not just for the joy of being up there and it being an amazing sight to take a photo, it's also to let your camera know that from then on you're in a new site and all the photos you're about to take of epiphytes are going to be in a new location. So as you move down, you take photos of this. This is a bird's nest fern. You try and take photos of the leaf itself, but also any characteristics that might help you to identify it later. In many cases, and with the more common epiphytes, we're able to take samples as well, which we can then compare with specimens and collections, but we want to do the least damage possible. And in fact, many epiphytes are also protected species, so you don't want to disturb them at all. So really taking photos of these are the spores, which are the primary identifying characteristic for ferns. That's really, really important stuff. This is a really nice fern that I particularly like because it's actually got 2 different shapes of leaf. So they're dimorphic. You've got the ones which are collecting debris, these brown things close to the trunk, and you've got the photosynthetic and the fertile ferns, which are the green bits coming out from those. So you've also got to make sure to take photos to ID both parts of it, rather than counting it as 2 different epiphytes when you're going down. This is a picture of several orchids. I really like it because it shows us that there are flowers. Because the tropics aren't seasonal as such, you've got to be really, really lucky to find any flowers at all. I think this is one of 2 flowering orchids I found during my entire field season. But if you can find them, they're absolutely amazing to allow us to identify them. And orchids are heavily protected, so there's no way you can take a sample unless you get some very, very good permits, but it would not have been worth getting for the scope of my master's thesis. So once you've done all that climbing, you're left with the joyous work that happens on computers, which again, I'm not going to go too far into, but having collected various different metrics of how to understand our trees, I created some minimum models looking at just the things that might have an impact, and eventually came up with these wonderful graphs that show the hugely shocking surprise conclusion that had already been found in Panama. Bigger trees have more things living on them. And it sounds really obvious, but it's very, very important to check that what happens on one side of the globe is also true on the other. And indeed, some other things didn't quite hold true. So there's ongoing debate about the effect of bark texture, for example, and we didn't find that on our site as being an important thing. So really, canopy work is this hugely varied and evolving technique, which has changed hugely over the years, over the decades, and is still kind of in its infancy. And it's going to be really exciting to see where it goes with new technologies, especially as drones evolve and become more complex. One of the main limitations to automating a lot of our work is we don't have a resolution. We can't get inside the branches to work things out. And it's very hard to take cuttings with a saw on top of something that's flying. There are people actively working on that right now, and I think it's really, really cool. But the main conclusions for this talk: epiphytes are diverse and they should absolutely be part of conservation planning. We need to know what things are doing in the canopy so that we can better protect the system as a whole. And we have to have ways that we can understand what's happening in the canopy, even when it's cost prohibitive to actually go up and check for the site that you're trying to protect. So understanding the hosts themselves can help us to prioritize where we actually want to do our canopy conservation, rather than necessarily needing to go up and check individual epiphytes all the time or at every site. And finally, I think my main conclusion from this presentation that I want to say is canopy science is really cool. So I'd love it if you have questions about it. I have many thanks to give. Obviously, the Seven Village, thank you very much for having me, but also all the people who helped work on this particular project and who have hosted me while I've been in the field doing canopy research. I think that's me, so I will stop screen sharing and potentially take questions. Thank you so much for listening.